Hello everyone. Assalamu alaikum. I hope you enjoyed this MMDR planning conference with multinational research experience as well. And this is the last session where you will experience the three dimensional structural modeling AI-driven approaches by our computational experts. I would welcome Professor Dr. Riazuddin, who is a professor in Institute of Chemistry and International Center of Chemical and Biological Sciences, and will lead us in the AI-driven approaches. So he will introduce you how the AI works in point. Sir, please.
Hey, uh, good afternoon, everyone. So I won't take much of your time. I would like you guys to do more into your laptops. So I would give you a brief idea about maybe if there is any slide. Okay. So uh, AI has been instrumental for last couple of years. Everyone is being affected by it, and in every field and every aspect of your life is basically affected by AI, whether it is your schedule in terms of calendar or your mobile phone, smartphone, and whatsoever. In the same way, AI impacted the scientific field. And one of the area where the AI has seen the maximum application is basically the field of biology, where bioinformatics is one of the cornerstone these days. And if you see the AlphaFold is mentioned here, and which has been given the prestigious Nobel Prize last year. And the idea is basically it gets the fundamental concept of AI from where it actually takes the pattern recognition from the sequence of the proteins. And then it was able to predict the structure. So protein structures are basically more conserved than the function. It is well understood from the molecular biology. And that's why AI has found this application in the area of structure prediction a little bit easier than the function prediction. And at the same time, every protein has dependent its structure on 3D folding, how well it is being folded actually. So sometimes the misfolding results in disease stage. For example, Alzheimer disease, if you have heard of, it's a famous example where a protein misfolding is basically resulting in the onset of Alzheimer disease. So AI-based structure prediction by AlphaFold is one of the greatest example AI has seen in the field of bioinformatics. And I think you will be getting a touch of how to use AlphaFold in this workshop today. And as I mentioned yesterday in my talk also, this can now easily be automated. And you can find there are lots of tools are available from where you can by using not much coding, you can easily use such kind of tool for prediction of your structure. And those structures are very well, I would say, validated by methods such as Ramachandran plot and others. So as long as you have sequence and you have some similarity, even if you do not have similarity with the template, you can even go for prediction with the AlphaFold by using the pattern of the folding for which the AlphaFold has been trained actually. So AlphaFold success is basically for the purpose of structure prediction basically lies in where it was able to solve the unknown protein structure even which was not being able to solve before. And millions of sequences are now being solved by AI. So with that, I would like to hand it over to Uruj again, or we can introduce the next speaker and then see how it goes. Thank you so much.
Thank you, sir. Thank you, Dr. Riaz, for a nice introduction about DA. Very quick. Next, I would like to introduce the teacher of Dr. Riaz. So, from pioneers of computational chemistry in Pakistan, Professor Dr. Zahir Al-Qasmi, who introduced computational chemistry and leading the computational chemistry in Pakistan so far. Sir, please, sir. Please say something. AI-driven approaches.
No, I'm not going to say AI-driven approaches. Assalamu alaikum and good evening. Well, it is an honor for me to see so many students sitting over here just to attend this workshop for the 2 hours. And I'm quite familiar with this field for the last 20 years. I know it very well. It cannot be understood in 2 hours of time. Then my real question is that why we gather over here for 2 hours? The simple answer is that because this is an introductory platform where we would like to see same-minded people who have interest or is going to be moved in this field. So this is our family meeting in this direction. So we are here. At least we are going to be familiar with each other. So this is not a 2 hours. This is a long bonding interaction. So we are going to be interact with each other whenever you are working in this field and need any kind of help. You can always approach me, my team. You can always approach Dr. Riyad, Dr. Ruth. Here they are going to be handled. So in next 2 hours, what you are going to learn, this is just a glimpse of the things that you are doing. So let me introduce ourselves of my field that we established this computational chemistry back in 2003, which was originated for me and a trade in Austria with Michael Rodelac. And then since 2003, now 2025, my lab is particularly working only in vivo. So we are working definitely only with computers. You hardly can find any lab like that, particularly in Pakistan and maybe in a similar culture. When we started at lab, it was almost impossible. That was the first lab. But now you can find there are a number of labs which is called what is the message? This is a field of science which can lead independently, can work closely with the experimental people, and our target is identical. We would like to explore something. And when as a scientist you would like to explore something, you can use any tool. And this is one of the tools. Basic understanding to explain it. So whenever you have any mystery, you would like to resolve something, computer is there to help you. And now with AI driven, the same thing which we are doing from the last 40, 50 years in this field. Now that everything is going to couple with the AI so that you can do even faster. And the purpose is that you would like to use more data to understand in a better way to give up a solution which we are looking for. So there's a long list of example where it remains unresolved by any experimental technique. Now we would like to resolve it. So if the single agenda, we would like to explore something as a scientist, and we are working on this. In next 2 months, you will have an hands-on experience, and that's why I encourage everyone to bring your own laptop, so you have a feeling that if you are guided, you can do and you can work in this field, you can contribute. I wish you that if you find interesting, try to find a lab where you can spend 6 months minimum or 1 year, so you can start working on it. I myself presented here is not a by birth chemist, computation chemist. I have an interest. I found the opportunity. Someone provided the opportunity. As a result, I grow over here. And then the thing is going on. Same for you. If you are, of course, if you are not by birth in computer guy, but still you would like to take the advantage, you have the right and you can do it. I can assure you. The only thing you need a dedication and time. This is not a short-term memory. This is not a short journey. You have to find it. Okay. So I hope the next 2 hours you will enjoy, and then we are going to have a small question answer session online. You have to do it just to make sure that you participate physically as well as intellectually. So you are not somewhere else for this. When you fill this form, then we will be in contact with each other and will remain in contact. I hope. Thank you. Yes, Manoj.
Thanks, Professor Abdul. Okay. So, Professor Abdul, I already mentioned you that this workshop will end on a quiz. So you can concentrate on workshops very well. And after the workshop, you can pass the quiz. You will be given an e-certificate as well on your WhatsApp account. Okay. So the content of this workshop, as you see, It's a protein because I know most of them pupils are not from the biological background, right? How many of you? Let's be more interactive. Okay. I'm from chemistry science, purely chemistry. So there is an overview for you, all of you, for the protein and its basic structure, followed by sequence and structures. How many kinds of structures are there present and what we can utilize in this workshop and for the AlphaFold specifically. And as Dr. Vyas said, that AlphaFold won the Nobel Prize last year, and it is based on AI-based predictions of structures, and it is very quick. Rather than conventional structure prediction approaches, AlphaFold gives you a fast prediction, and it is on your choice that if you go for a comparative modeling or a template based or an ab initio modeling. So it gives you a chance of whole three options, and this prediction will give you a model, and which you can analyze or evaluate through different servers. That is, two servers are mentioned here. There are a number of servers, but these are the most reliable servers who can give you an instant results. That is PROSA and SAFES. And afterwards, as we know that number of our research objectives is linked to mutational analysis. Number of diseases like non-synonymous single nucleotide polymorphism end up in the mutations in the genetic system. So we give you a little bit glimpse of how the mutation analysis can work on this structure. So if you develop a model, you can analyze its mutations field as well. So in the end of the workshop, you have a little bit glimpse of mutation analysis as well. I will like to welcome Ms. Atia, our senior in PhD. So she will explain you the protein AI-based AlphaFold, how to do this. And in the last, I will explain you the dynamic analysis. So Ms. Atia, please come.
You can hear? Okay, before starting the workshop, I would like to ask a few of the questions related to the workshop that you have been in the group, on the WhatsApp group, all, right? So there are some people who are not in the WhatsApp. So we actually shared our workshop manual in that WhatsApp group, which you have, if you have that, so you can download it and you can, if you miss any steps, so you can go back or move forward. So that will be an easier way.
Okay, I'm getting this USB. You can save the workshop manual. So from this.
So I would like to know that how many of you people who does not have that manual, workshop manual. So a USB is being rotated meanwhile. So you can just copy that manual in your laptops and then open that PDF and follow up. That manual is also shown here. So you can follow from here until you get the manual. In the WhatsApp group, you are in the WhatsApp group? Good. You did not share the manual.
Okay. First thing, are you the listed participants? How many of you are in the list which we shared in the WhatsApp account?
Yeah.
WhatsApp group. Okay, I'm sharing it again. You are not in the WhatsApp group. I'm not in the WhatsApp group. You are there.
I'm not in the group.
So I'm sharing this USB. There is a folder for workshop 2025. You can save it in your
Okay, so I would like to tell you all that there is no such kind of any
Check your email, check your internet if it's working in your laptop. And if it's working, that's fine. So we can move forward. Actually, you can start and meanwhile they can
So all of you who hasn't got the manual
yeah, sure.
can see it in the screen. The manual is there. You can follow from here as well. It's not so kind of difficult thing. So there are some just links which you, if you will write it in the Google, you can also open those websites and web servers as well. So at first, in the starting of the this workshop, I would like to introduce a few of the points regarding the proteins. That proteins are actually the large biomolecules and macromolecules, which you all, maybe you are all familiar with the term protein. And they are the biological macromolecules, which are designated for a specific function. They have a specific structure, and they perform various functions within the organism, such as metabolic reactions, DNA replication. Responding to this stimuli, protein may differ primarily in their sequence in the amino acid. You all know, if you are all familiar with the proteins, you all know the amino acids as well. So there are kind of 22 amino acids which are repeating in every protein, but the sequence is different, so the protein becomes different. And if the protein becomes different, their structure and the orientation, everything comes to the change. So to have the knowledge for the three-dimensional structure, it helps in the rational design of the drugs. So you can have the structural information through the experimental techniques as well. The X-ray crystallography and NMR resolution works also for the structural experimentation. But what if you are unable to identify or to determine the structure three-dimensionally? So you can opt to the computational approaches for this, because there are some proteins such as disordered proteins, the very well-known proteins in our biological system, which still the experimental experiments are not able to resolve their three-dimensional structures. So for this, we use computational driven approaches. So from sequence to structure, we first talk about the primary sequence, which are composed of the one-dimensional sequence of amino acids. These one-dimensional amino acids form the secondary structure in the beta sheets, alpha helices, and then these all secondary structure merge up, make an orientation, and orients itself in a tertiary structure, which is also known as the third degree structure, and then the multiple third degree structure or tertiary structures come together and form the quaternary structure. So for the, as I told you all before, that for determining the structure, you have experimental techniques like NMR, X-ray diffraction, and electron microscopy. But we here will talk about the in silico structure predictions for protein. So computational method for the protein structure prediction are divided into two classes: comparative modeling and ab initio modeling. For example, if you have a template before, like you know that how this structure would somehow look, you have a template. We call it template in computational terms. So you go to the towards the approach of comparative modeling. But there are some cases where you don't have the template or any information regarding the three-dimensional structure of that protein. So then you decide to go to the route of ab initio modeling. It is also known as the de novo protein modeling. So for the template base, this is the pipeline of the template-based modeling. For this, we retrieve the protein sequence. We have the one-dimensional protein amino acid sequence, and then we go towards the template recognition. Okay, so template recognition, multiple sequence alignment, and then we go towards the model generation. And then after which we generate the model, we evaluate that model under different matrices, that how the model is being made. But here we are not going to know the steps about the comparative modeling. Here we will discuss about the AI-driven approaches for the protein structure determination. Why are we going to do that? If anybody has an idea, I told you earlier that there is a comparative modeling process. We have the template. So why are we going to, use the AlphaFold AI-driven approach? Why are we going there? Okay. We go to this route. As I told you, it is an ab initio approach. You can go you can use AlphaFold for both of the approaches, for the comparative modeling as well, for the ab initio as well. So by latest, you all know about how the variety of the functions are there for the AlphaFold. Shall we start our?
Yes.
Okay. So at first, we will first retrieve our sequence from the UniProt database. This is a database where you can find almost all of the protein one-dimensional structure, means the amino acids of the protein available. Okay, one-dimensional structure. Now you, from this structure or from this sequence, you will model three-dimensional structure using the AlphaFold server. So will you please open this website, www.uniprot.org. If you have opened this website, you will be drawn to a page like this. Everybody? You will be drawn to a page where you can write down the name of your protein. For example, the example which we are going to use here in this workshop is tryptophan synthase. It is a protein. We are using this protein as an example in our workshop. In case you are working on your own, you can write down the name of your desired protein which you want to model. Everybody, please write down this name in the search option. Yeah. Those who have the manual can directly click on this link given here, provided here. You can see it in the screen. And those who are not having the manual, can you write it down here? You will be driven to this page like this. And then in the search bar, you will write down the tryptophan synthase in your.
Can you share the symbol with your WhatsApp group again?
Yeah, sure.
Yes, sure.
Okay. I would like to know how many of you have already searched this protein. Just raise the hands so that I may know the number. Okay, thank you.
Next step, well, you have to finish in a proper time, In this way, we cannot finish it in 2 hours. And then it will be very difficult, and if you will not reach the final destination, you will always remain thirsty. So be fast and quick, okay? Better to move in the next step.
At any step, if you have any kind of query, you can ask at the end of the
raise your
session. You
hand
can just
and
write
try
us
to
the questions
Yes.
for us, and we will try to answer your question at the end of the session.
Okay.
So after this, when you will press the enter, you will see such results, some kind of results like this here. Here in this database, you will find all of the annotated one-dimensional structure, means the protein sequences in this database from all of the species which have been identified. For example, the tryptophan synthase in the Aspergillus, tryptophan synthase from Escherichia coli and the Salmonella typhi as well. These all, are the proteins, different proteins from the isolated from different species. Many of them would be same, but you can find it specifically for each of these species. So you can work your desired protein, your desired species from this database. You can take that and do your stuff whatever you want. So here in our example, we will be taking the tryptophan synthase from Escherichia coli. You can just click on this link here. This is the step 3. If you click here, you will see a page like this. How many of you are on this page? Yes. Okay. So this page shows you the exact identified information about this protein, about the tryptophan synthase from E. coli. You can here see the protein name, the gene tag, the status, the organism. From here, there is different slots. You can know the function name, taxonomy. In our modeling approach, we are directly going to click on the sequence section here. When you will click on the sequence section, you will be dragged down to a window like this. There you will see all the amino acids present in the with the numbers as well. All the amino acids present in the protein. Okay. How many of you are on this? Okay. Now on the step 5, there are 2 things you can do. From the cursor, you can directly copy the sequence from here as well, or you can directly just click on this copy sequence, and all of the sequence will be copied, regardless how many amino acids are there. So once you will click, you will see this copy sequence from copy sequence to copied. Okay.
You guys will be just looking different scale.
Okay, so you have copied the sequence, right? I would like to tell you just a point here that when you copy the sequence and move towards any window and you do some function, you will have to copy it again. So when you are ready, all ready to go to things, the website you want to go, then come back, copy here, and then paste it down there. So you have to just do it directly. We have to do that first time. Yeah. So first you have to copy it here. Then just open this alpha, just write down on the Google AlphaFold collab here. And you will see this Google collab actually. And in the Google collab, AlphaFold has already given the all of the its code publicly, where you can modify it, play with it, and make your own structure. And once you click it down here, you will see a Google notebook, collab notebook. Will open. Okay, it will be linked to your Gmail or any kind of email which you have been using in your laptop. You have to, if you are not signed in, so you have to sign it for that, for this. Okay, how many of you have opened this Google Colab? Now, I would like to break down the Google Colab notebook for you to have better understanding that in the very first few lines, you would be shown a cell which was named from for the input protein sequence. Are you able to see that in your own notebooks? Okay. So here, what this cell does is, this is your query option. So whatever protein you are trying to model, you just have to copy that sequence which you have copied it earlier, and then paste it here. This is an example. So you have to erase it and then paste your own sequence which you have copied earlier. First, when you will paste your sequence, then you need to run this cell. So for running this cell, you will see a play button here. Okay, when you will press it, the cell will start running. And once it is complete, you will see the result down there and also a tick mark here. First, paste your own sequence and then just play the cell. Okay, in this cell, you can also change your job name, any name you want. Okay, number of relaxation. So this is the cell where you want your models to get relaxed multiple times, 0 times. You do not want your model to relax, means you do not want your model to be energetically minimized, or you just want such a model. So for just an example, we are using the 0 here. You can use any number.
So you are done with step 8. Yeah. You are already completed step. Just raise your hand if you are not done 8 step. It is very difficult, you know. If we no, it's not a. Basically, the problem is that if we individually will do this, we cannot. Now you have to get help from your next. So then to reach to that decision, and then we can move. Otherwise, we can never finish it. Please, can you please help us? Quickly, so you can just give 1 minute break. So everyone completed step 8, and then we will move.
Yeah, we are.
Now talk with your next neighbor.
Okay, I'm going to tell you this. Once you see the tick mark, it means your cell has been run.
Otherwise, if it is a long way, then if you pick one, you cannot try the next one. As a result, you will cut. Take the advantage of the neighbor and then fix it. Okay. Now we will move to the step 9. Chale.
Okay. So all of you, those who have played this button, would have been received the tick mark. Some of you might be having the tick mark in the 0 seconds. It's actually the internet strength. It depends upon the internet strength that how many time it takes to run a single cell. So this is
So once you have run the cell from here up, you can see the result. You see the job name you provided, you see the sequence you provided, and you know the length of the sequence. It is giving you the result that whatever the sequence you provided, its length is this, and you provided the job name this. Okay, now to run the AlphaFold, you have to install the dependencies like the all of the codings which AlphaFold server uses in the Google Colab. So at first, what you have to do, you have to first play the install dependencies first cell here, where you can see the cursor. If you're seeing the mouse here, just play this button. Okay. Now, the next option is that MSA sequence, multi-sequence alignment. So AlphaFold using its own directory or the directories associated with the AlphaFold server uses its template to model your desired protein. So when you will select the MSA option MM sequence 2, UniRef, it by default takes its own template, best template to model your best structure, best possible structure. You have to just click on this and then pair mode. In the pair mode, unpaired paired. So it just do the template alignment unpaired wise and paired wise. Whatever model it takes, it first aligns it to that how many percent identity is there. The person, depending on the person identity, it takes the best template from here and will move on to the next step. So first you have to play this button to install the dependency, and then you will have to just, if any other option is selected, kindly select these two options in your mode. Okay, and then play this button until you get the tick option. At first, you have to install the dependency. It will show some of the things, collab running, some kind of options like this. When you get the tick mark, then you go on to the next cell, play it down until you receive the tick mark. How many of you are on the MSA options? In the next step, there will be a cell called advanced settings. Are you able to see this? In the advanced setting, you can see the explanation of the option down there as well. So at first, the option is model type auto. So if you select the auto option, it will use the AlphaFold for monomer prediction. Okay. So if you know the proteins, you know that proteins exist in the monomeric form, homodimeric form, heterodimeric form, oligomers. There are so many kind of domains. So whatever function you want, whatever kind of protein you want, you have to select that option. So in our case, we just want a simple protein, no homomer, no heterodimer, no kind of oligomer or any kind of other thing. So we are just trying to use the auto option. In your case, if you want to model an oligomer, so you will follow the option like this, AlphaFold to multimer V3 for the complex prediction or for the monomer or oligomers. You can select as per your desire. Okay, so here we are just going to use the auto function. Then in the num recycles, okay, I would like to tell you one more thing. You do not have to run this cell right after you click one thing. This whole cell is one. So you have to first select all the options you desire, and then you will press the play button. Okay. So I'm just trying to break down the all of the information present in the cell for you, so that next time you will do your own or independent work, you will know what is what. Okay. So in the num recycles, okay. So if you are listening me, when your AlphaFold uses the template you have provided the MSA alignment. You have played that button, right? It has utilized its own template available in the database. Now, it is trying to generate the model. To generate the model, you are using advanced techniques of the AlphaFold server. In these advanced techniques, what you are trying to do is you are trying to model a protein. But before having a structure, before having a three-dimensional structure, what you are trying to get is that best possible structure using the advanced technique. Okay, so it's just like a refinement of the thing. For example, whenever you purchase a thing, you just try to find the best possible version of that, right? So we are just trying to do right here. We are just trying to do that as well. So we will be able to have the model if we do not do go on the advanced setting, but to have the best possible structure, we need to look onto the advanced setting here as well. So in the advanced setting, when you selected the auto, now it knows that you are just wanting an only the protein three-dimensional structure. No heteromer, no complex, no oligomer. Okay. Then in the next step, what are you trying to do here? Number of recycles from auto. Okay, if you will press it down here, you will see various numbers, 1, 2, 4, 5, maybe 5 cycles. So what you are trying to say here, the AlphaFold server, that then generate the model. Generate it in auto. You can see here, select num cycles 20. It will generate the model 20 times. 1, then generate again. 2, generate again. 3, generate again. And find the best orientation of your protein. Okay, if you use higher number, remember this, computationally it will take more time. Okay, so if you want to do something quick, you can less the number, accuracy will be compromised, of course. But if you want to make sure that the quality is very good, you can select the auto or give your own number as well. So here, do not use the auto here, just use the least possible number so that you can run on the expected time. Yeah, you can use 1. Okay. Okay, so in the next bar, you see relax max iteration. Okay, whatever number you have given, for example, you have given 1. So again, the recycle will be 1, but its iteration means it will repeat itself 200 times. This is all to make your structure more accurate actually. It should be done auto for actually it's the tolerance means it is the threshold for the errors of the model. So you for this auto you are going to say that to the AlphaFold that only bear the error of 0.5, just 0.5, not less, not more. Okay, the auto suggests the AlphaFold that you accept the model which have the error frequency of 0.5. And then it will repeat itself 200 times. The iteration will be 200 times, and then the model will be generated.
Remember, everything depends on the how much accurate we are looking forward. Since over here, right now, we are not looking forward to accuracy, we are just trying to learn the step. So we keep everything in a minimum way so we can move forward. And when you are trying to do by yourself, then you have to do it accordingly, what were your requirements. So 10 steps you already done.
Okay, before going to the next step, we have encountered some issues regarding the collab. So some of you are unable to install the dependency step, right? So for those people who are unable to install the dependencies and follow the tutorial, an announcement I would like to make that we will provide you the results of this. Okay, we will provide you a zip file for this at the end of this collab, so you can move on to the next one. Actually, all of you people are using the internet, so the internet is slowed down depending upon the hardware you are using.
So it's absolutely fine. Just focus on the steps. So
Yes.
then you can learn from that step, next step. Because they are not dependent on each other. If you have the result, you can move in the next step. Okay?
If you have any query, just write it down. If you will listen it carefully, you will be able to do it on your own in the later time. And if you have any questions, you all have the email addresses of Sir, mine, and Dr. Ruj as well. So if you have any query, you can question it down to us at your email at our emails as well, and or at the end of this session. So in the next step, pairing strategy. The word you can see here, greedy. All of you are able to see the word greedy. Greedy, what does it mean? It means just follow my, it's the simple, in simple words, you can say that follow my instructions, whatever I have given. Become greedy. So here it's also for biologically greedy means the pair any taxonomically matching subset. So it will find any match from the database and will make your model. It will become greedy. And then you do not have to check the extra PTM. You may check this if you have enough time, enough hardware, okay, enough resources, you can check it, but here we do not have time. So for the shortage of time, we are skipping this. Is that? Okay. Now, after all this, if you know, if you have followed the manual, you see the options. Options are, if you are matching with the options, just play down the button. Now, advanced settings. This one. No, you do not have to do that actually because it will save your basically you can do that when you will do your independent works. You would want to save that in your Google Drive as well. So right now here you do not have to just take it down because in the last cell all of the files will automatically be downloaded. So you do not have to save it here. So if you have played this button, it will take some time, maybe. Yeah. Yeah, it will take some time. Actually, it gives you, for the better accuracy, the options are limited. So the least iterations you can select are 200. You can move on to the 20,000 as well. Yeah, the tick means done. If you receive the tick mark, it's done. So it may require some time if you have selected this option. How many of you have received the tick mark for this one? Okay. It means your laptop is good. Okay. So here the question you asked, the lady who asked the question that you can select save all, save recycle, the recycles. You can save all of the models. You can save all of the recycle. You can save all the data to your Google Drive if you have enough space. Okay, DPI it is the resolution of the structure and the picture quality. So whatever DPI you want, for example, by default, it's giving you 200. You can move to 300, 600 for the better resolution. Just click on, just leave it in the 200. So because you do not have to publish this work. Okay, and then run this cell. Run this cell, save settings. So whatever, it means that whatever settings you provided, it's going to save it. And then in the next step, if you want, listen here. In the next step, the run prediction. Now it is going to run your and generate your model. So you can check the option here or uncheck it. By checking on this option, it will run the code and will be shown to you on the screen. Okay. But if you uncheck this option, it will run in the backend. So some of you people might want to see what is going on, how are my early results coming up. So you would want to check on this. And if you do not want to see any kind of information on your screen, you may just leave it unchecked. So depending on your interest, just check or uncheck and then run the cell. But I would like to like you to know that when you will check on the display images, it will take more time because it will run and then will show it to you as well. Okay. And just a little information that you do not have to you can say the surely check this option, because whatever the images you will be shown in your window will be downloaded in your results as well. Okay. So if you just want your results, you can leave it unchecked and see it at the last. Or if you want to know that, okay, I don't think it's good result, it is the good result. If you are able to analyze that, so you will just cancel the whole thing. That my model is not good. We'll show it in the graph as well. If you know the validation protocols. How many of you have run this cell and get the tick mark? It's running. Okay, those who have checked may require some extra time. For those who have checked the display images as well, so it will take a bit of time. Those who have received the tick mark can go to the display 3D structure. Okay, in here, just look at here. Rank number one means the top rank model will be displayed here. Okay, if you select 2, the second rank, third rank, 4, 5th, provided the number you provided the up on the upper cells. Okay, play it down. It will be shown images like this for before. Try some kind of results like this. Then you will the display. You will get this structure. Shall I play it again? If you check on this option and play it down, it will show results like this down in the cell. Okay, then it will run. At the end of the cell, depending upon how many seeds you took, how many recycles you provided, it will show the results like this. And then when it's get completed, you will see the last line will be done. And then after this, you will play the display 3D structure cell. Once you get the done line, then you are going to play next cell. Show side chain. This is for the picture. If you want to have side chains as well in your picture, just click on these or options. How many of you are on this display 3D structure? Actually, the reason it takes time is that by the end of this cell, your results are completed. Your model has been generated. Now, in the next step will be all of the plots you will need the validation from the AlphaFold. You will get the plots and you will get the zip file at the end. So from here till here, you are having the three-dimensional structure of your protein actually. No, there are few steps left. Yeah. If you have received the check, the tick mark before the previous cell, we can move on to the next. Just make sure that you check the lesser amount of the display result because it will take more time. Results provide prediction on हमारा last step था one model provide तो अपना लंबा चाहिए 1% कर लेना Okay, for those people who are unable to utilize the cells and the AlphaFold are unable to get the results, the zip file of the results are being shared. Okay, so you can left it running, you can leave it running. It will run, will get your result. You can compare the results we provided you and the ones you get. Okay. You don't trust. Actually, so many people are using the internet facilities at the same time. So, depending on the hardware, it sucks. For those who have completed the previous cells, the display 3D, the plots. Okay, it's still running. So if you will just get the tick mark for the display 3D, you will get this kind of display. I'm just telling you this because once it pops up on your window, you may identify that okay, this is done. After you get this three-dimensional structure, you will then play the cell plots. Okay, as we are running out of time, I'm going to display the results for you. You will get the results in the WhatsApp group as well. Those who are in the WhatsApp group will receive the files, result files, the PDB files, and everything. Meanwhile, you are running your own structure prediction. Once it will be done, it will be shown in your screen as well. So you just have to keep up with the results that how you are going to analyze it. Okay. If you just check on this plots, you will see the plots down there. Now, this is the last cell which you will run in order to get your results. Package and results download. So when you will play this button, this is the last step of the AlphaFold. That when you will play this button, it will give you a zip file. Okay, it will be a compressed file, and all of your results, plots, models, how many ranks you have provided, recycle seeds, everything will be provided in that file. And here I have placed a screenshot of that folder as well, that when you will decompress that folder, the zip file, it will be shown like this. For example, you have provided the name of your job was, my name was job, test. My job name was test. So the initial names are test. You are able to see the display files, PDB files, but these are the structure files. The PDB file is the file you require. So you will get the PDB file for the different ranks. And I would like to know what rank should we select for moving on to the next step. If you know model number 1, 2, 3, or 4, or 5, we have 5 models. Suppose what model should we select to the next step? Means what model should I take that it is good? No. 1. Yes, right. 1. 1 means the best result. Number 1 will be the top rank model. Number 2 will be the second, third, fourth, and the fifth. Depending upon how many numbers you have provided, it will give you the models for that. Even 10, 20, 50, how many you provided, depending upon the numbers you have given in the data. These are the plots you are going to get from the AlphaFold server. The AlphaFold gives you its own validation as well, that whatever model it has generated on the matrices of AlphaFold, how good that model is. Why I am saying that this is the rank number 1 model, this is the rank number 2 model, this is 3 and 4. So you can see it here from around how many sequences? It utilized 10,000. 10,000 sequences out of 10,000 templates. Your model was built. Okay. This is the local errors. If you just split it down here, the result, if you analyze this result, the blue means 0. The lower the number, the lower the error. The lower the number, the lower the error. Showing that your model is good. For the rank 1, 2, 3, 4, 5. Now, the best one you can select. So ultimately, by itself, it has given you that this rank 1 model among all of these scores, the rank number 1 model is the best one. So you just repeat. Because according to all of these scores, this score, this one, and this whole one, the AlphaFold is providing you the information that the rank number 1 model is the best model among all which have been generated. So by default, all of the models you get, you select the rank number 1. It shows you that it is the best model generated among all. अच्छा वो scores का मैं बता रही थी आपको कि जो graph है, the graph is showing you the scores here. The less than the number, the bluer the color, means the good your rank model is. Okay. In our case, All of the models' qualities are good, but the best one is one.
Why? Why? Sequel to false.
Because we selected an example in such a way that it will give you lesser errors and you can run it smoothly. That's why. But it might be not in the case if in your desired routine, results might change depending upon the availability of the templates and in the database. Okay. Of course, for every step there are some limitations. So, okay, there is another solution for this as well. We have provided in the manual that if you are unable to run the AlphaFold using Google Colab, you can use AlphaFold server as well. But of course, the limitation here is that you cannot play with the cells like the way you did before. Like you can't select the recycles, you can't select the seeds, you can't select the number of models generated. You will be dependent upon the server of the AlphaFold. So here you just provide your sequence and then model. The model will be generated to you.
Remember one thing, in almost all kind of software, whatever you are doing, normally they come up with two different options. One is called stand alone, means you install everything in your own machine and try to change whatever the parameters depending on your own choice to improve a result. On the second, it is called by default. So by default means they have already practiced it and which is the best they have already made it. So you have no choice to change anything. Normally when we do something by server, server is always kept as a by default and you submit your result, your input, they will come output and you rely on it. If you are an expert and you would like to do something good, it is always advisable to do external machine. You install yourself and your own machine and play by yourself. Okay? So do you guys need a relax 5 minutes? Or is okay? That's lovely, cool. I'm really happy. Normally, I have a quite long experience. Whenever you have a session like that, this is quite normal. The few of them cannot follow you. And as a result, all the program is hacked and nothing can be done move forward. But I can see from the back that most of the system is following the thing. That's very
Okay.
very good at me.
Thank you, sir. For this, to encounter the problem you are facing, all of you just to have your own, just to follow the rest of the things in the manual. What you can do is you keep your AlphaFold on run. Okay, leave it there, and use that sequence from the UniProt database, and you may use to model your fixes from the server as well. You will get the model from here as well. You can download the results and can move on to the next step as well. Okay. So those who are unable to run the Google Colab right now to this server thing can use this AlphaFold server and get your results so that you can follow the next steps as well. Right. Who this? Okay. Those who have got the files, how many of you have got the files?
All files.
Okay, in those files you have seen many PDB format. There are 5. So when I ran that AlphaFold for you, myself, I selected I said it to generate 5 top models. Okay, 5 top model. Out of 5, the best model is 1. But in those files, what you are seeing, you select the type. Okay, there are so many PDBs. Relaxed, unrelaxed. These are all the results. I told you earlier that you will get all of the results. The one you provided earlier, the one you provided later. Unrelaxed are the models which are not relaxed, which are not recycled. No? So you will select the model number 1, relaxed, rank 1 model. Relaxed. The one energetically minimized. Okay. This one. 001. Sorry. When I provided you the results, I renamed the top one model as the model number 1. This model 1. This is the ranked 1 model. Actually, for the sake of, to get the best result, I just renamed the file so that when you select in the next steps, you don't select the wrong PDBs. This model 1 is the best model among all of the PDBs. Its name used to be like this. So for the sake of better visuals, I just renamed it as model 1. Okay. Model 1. Model 1 and model are the same PDBs actually. They are the same. It just got copied 2 times. What? Which one is not working? It won't open. It won't open. Okay. Here, listen. If you are trying to open that file, if you are trying to look upon the information of the file, just do this. Because you do not have the visualizer. Just remember this. You do not have the visualizers for the protein. The software to visualize the protein, to have the complete structure shown, the AlphaFold, the picture like AlphaFold showed you in the previous result. Here you can see this one. To get this picture, you are trying to get this picture. Yes. You will not get this picture from the PDB file actually. What you have to do actually, you have to get a visualizer for this. You open that PDB in that. For example, the very well-known software is the Discovery Studio Visualizer, DSV, also known as the DSV or Autodoc. These are some kind of softwares which gives you you can say freedom to open the PDB files. Okay. So in your case, if you do not have these softwares in your laptop, you will be unable to open PDB. But just to know that your PDB is fine. Sorry. I'm trying to go to the internet. ये भी किया था मैंने. Okay, हो गया. ठीक है. ये था ना? नहीं. आपने वो फाइल ओहो. If you are just trying to know that what is the information provided in your file, you can open that file, the model 1, with Notepad. You will be able to see the information in there. Okay. I'm opening here itself. See this? Are you seeing this? When you open your PDB file with Notebook, this will give you an information like this. It gives you the PDB file is the complete coordinate file of your proteins. Means every atom present in your PDB file have a coordinates in three-dimensional space. Means where should it be placed in 3D. So, for example, methionine, the residue number 1 was methionine, M. You can even compare it with the sequence you have copied from the UniProt as well. That the number 1 residue is methionine, and in methionine, all of the atoms, for example, nitrogen, hydrogen, 3 hydrogen, carbon, which hydrogen is where? These are the coordinates. Okay. So when you will open this file in any visualizer, it will give you the complete structure for this. So this is the PDB file you get. And if you want to know that your PDB file is okay, you just open it with a notepad and just see it that, okay, There are the residues, complete residues are there. For example, I had about okay, listen. I had about 397 residues, right? So all of these residues are modeled. They have coordinates, atoms are there. This is just, you can say, shorthand way to know that your file is okay. Okay, now all of you please listen. Now I expect that many of the people, not all, but many of the people present here might have got the files ready. So we are going to move on to the next step. So the next step is validation of the model. Why do we need to validate our model? You know, if I do a work, for example, here, standing here in front of you, I'm trying to make sure that you follow the manual, okay? If somebody asks me how was the workshop, I would say it was good. This is my internal evaluation. I would say it is good, it was good. But based on your experience, you will say how you felt. Was it good? Was it worse or was it not? This is external validation. So, in your opinion, which one is good? My internal one or your external one? Hybrid. But most reliably, if a third person will listen, you would say she has conducted her workshop by herself. She is evaluating herself. She will always say, I was good. But what people are saying is more valuable. What others are saying, because those results are unbiased. Okay. So the validation matrices which I have shown you earlier was the one provided by AlphaFold server. The server which used its own template, its own database, its own coding, showed you generated you, give you the best possible results as said by the server itself. Okay. It provides you the validation as well. Okay, this is our model. This is the error. This is the plot. It's good. You have seen that these models are good. But is it really good? You have to do or validate the model externally. No? To have unbiased opinion or unbiased result. So you can publish and say to the reviewer or in any journal that, okay, I have generated this model by AlphaFold, validated its model by AlphaFold and in the external softwares as well. And it passed both of these. Okay. So now you know the purpose for the external validation of the models? Okay. How many of you has got the idea of external validation of the models? You understand this, right? Okay. So we should move on to the next one. There are many groups who are working computationally. Okay. Day and night, you may say. And there are so many groups who develop many kind of web servers, many kind of websites. open source, licensed. You have seen, all of you may be familiar with the molecular docking as well. So for the molecular docking, you have seen so many versions, Autodock, free, available freely. Okay, there is Schrödinger as well, licensed, very expensive. Then there is MOE, licensed but less expensive than Schrödinger, provided the functions, okay, provided the groups who develop those. Okay, you all got that. You can perform same experiment, the docking experiment, using different servers. But what you will do? Will you use all? Why? You will see or check what is good for you. Do you have enough resources to buy Schrödinger? Then use Schrödinger. You don't have any kind of resource, financial resource to perform docking. So you will go on the free option. Okay. Then if you go somewhere in any lab who has MOE, so you will run that experiment in MOE. Depending upon the hardware, depending upon the resources, you selected your version. Okay. Now, there is another criteria as well. When you select a degree from any university or you purchase anything from anywhere, you see its worth as well. How much accredited that university or that brand is. You know, some people, some group have established their names. That, okay, whatever work they do, it's well known. So reliability is all you can have. So for the sake of reliability, it means you are trying to evaluate the model externally. So you have to make sure that whatever web server you are trying to use should be reliable enough that provide you the unbiased results. Okay. So whatever means, I want to say that for whatever reason you have generated your model and now you are trying to validate it externally, you will find many web servers, many websites, many softwares which will give you the option to validate your structure. But what you are going to use is the one which are the most reliable. So among all of the validating web servers, the two web servers are the most reliable web servers, universal, globally used, you can say, for the evaluation of the models. The one is PROSA and the one is SAVES. The name was provided in the content slide as well for both of the servers. You can use both. Here we are just going to use one. You can, but I will show it to you. Also, if we have enough time, I will show the next one as well. Okay, so here, if you have the PDB, you just for this server, for the validation, you just need to have PDB file. It is the complete model of the protein. Now you are trying to validate it one by one. Rank number 1, rank number 2, rank number 3, rank number 4. Okay, the AlphaFold told you that the rank number 1 model is the best. Rank number 5 model is the least accurate among all. Okay. Externally, you are going to evaluate and judge by yourself. Is it really the case? Number 1, number 2, number 3, number 4, you are going to independently process it, validate it, see these scores, judge it, and then see, okay, yes, the AlphaFold was right or was wrong. Okay. So you will just open the link of the PROSA web. This is the one. You will write on the Google PROSA, and then you will open this website. Okay. You open this. This will be the interface. Are you able to see that? Are you able to open this?
Yeah.
All of you? I'm not getting the enough response actually, so I'm unable to judge whether you are on the same page or not. Okay. So there will be some options.
Okay, I'll break it down for you. So if you work independently, you may know that what you can do with this server as well. This is the first option. Please upload a structure in the PDB format. For that, this is the case if you model a protein by yourself, because you have the model. So you provide it to the server, upload it here, and then get the result. Okay. Or if you already have a structure, for example, for many of the proteins, you already have a structure. You do not need to model that. So for this, what you can do, you can directly write down the PDB code, its chain ID, or its model number. But in our case, we are not going to use this because we have modeled our protein by ourselves. So we are going to just upload it and then go click on the analyze. When you will upload it, you will see the file, your file name here. It means your file is uploaded on the server, and then you will run the button analyze. Okay. Now, PDB file. Just select the PDB file. Okay, I am doing here. ये वो का है Firefox. So listen here. If you are watching it on the screen, when I just click on the browse button, it opened me a window. From here, I can select my model. For example, I'm selecting the model 1. Okay, this PDB file I have selected. Open it. It shows here. Okay, and then analyze it. The page will be reloaded. See, it's running. Now it has been done. Now I'm just going to demonstrate how you can say that your results are good or bad. Okay. It gives you some kind of plots. The first plot is overall quality model. It gives you the Z-score. For the publication, you use this score, -10. From here, you can also see the black dot here. This black dot represents that among all of the experimentally identified proteins. For example, you know, in my earlier slides, I told you that there are many methods to determine the structure experimentally through the NMR and X-ray crystallography. For every structure, identified structure, you get a Z-score. Okay, this is the model quality score. The Z-score is also known as the model quality score. The more in the negative, the more the good. Okay, so here, this is the data already plotted for the experimentally identified protein Z-scores. The blue one, dark blue one is for the NMR. The light one is for X-ray. And your model lies here, -10. Okay, this shows that it is not an outlier, it's a good model, and it lies within the scores of the experimentally identified protein. So it is acceptable. Okay.
More negative, more reliable model.
Yes. Then the local model quality. Energetically, how much your protein is good.
how much energetically minimized your protein is. So if you have the this green line in the positive region, more peaks in the positive region or in the baseline, it means that your model is in the base. If it is in the baseline, it means your model is on the average quality. If it is in the positive region, it means that your model is not up to the mark. Your model quality is compromised. Okay. So if the local model quality values are within the negative regions, the more peaks in the negative region, the more good your model will be. Okay. And this is a representation that from every amino acid, it is showing its own lowest energy and highest energy. So from here as well, visually, you can see that which amino acid or which region is more energetically stable, means in the bluer region means lowest energy, means the stable model. And the red one displays low quality, higher energy, less stable. So the regions in the red ones are less stable, the regions in the blue ones are. So obviously protein is flexible. Okay. So you will always have some red regions, some blue regions. But how will you justify or identify that the blue, for example, your model is good or not? Means depending upon how many blue regions are there. There should be lesser red region than the bluer ones. Okay. If the more the blue, the protein appears to be more blue, means your overall model quality is good. It's energetically minimized. You will never ever in your life will see any protein completely blue because it's flexible. Whenever there is loop, there are turns, it will appear to be red. But of course, it should be lesser in number. Okay.
Okay, thanks, Miss Athiya. How many of you have can do it again alone? How many? Can you repeat it again? Or you want to? Yes? Not alone. Yes? So we can say this workshop is successful or not. Those who don't have don't members of that WhatsApp group, I can add them in few hours. Okay. So no need to worry about the files and or the e-certificate. There is a quiz I'm sending you in the WhatsApp group as well. We have to answer that, and those who have passed only can get the e-certificate. Okay.
We are not on the WhatsApp.
I'm taking all of your numbers or sheet. I will add you and give you the MCQ file. Yes. The sheet is rotating, you can write it. Okay. So you want to revise for once? For once, you want to revise? In the WhatsApp. Yes. So first we have copy. Okay, I'm revising it or summarizing the workshop. First we have copied the sequence. Whatever target you want to model, if you do not find any PDB or crystal structure in the data bank, you have to copy the sequence from the Uniprot websites. First step done. Copy the sequence. Okay. Then paste that sequence into the Google Colab of AlphaFold. Okay. One problem occurring in most of you is that the Python version of your laptop and the Google Cloud is not synchronized well. And second problem is that your graphic card of laptop is not enough to handle the display images of the Google Cloud. So no need to worry about this. You can use the AlphaFold server instead of Collab. Okay? So Adya already said that AlphaFold server has limitation. That's why we use Collab. Collab is a more advanced version and you can use more iteration limits or more model limits in that. But you can use AlphaFold server as well and report it in the reputed journals, international journals as well. Okay? So you copy the sequence from the Uniprot, paste it into the AlphaFold server or the Collab, your choice, your laptop choice. Okay. Then you run all the cells one by one. Remember that when you are working on the Collab, the first cell gives you the tick, green tick. Only when you will be able to run the next cell. Do not proceed the cells further once you don't have the green tick. Okay? Secondly, your internet connection should be very well. If it is not proper, you can't run the cells. Okay? So keep refreshing the Google Colab. For that, if your internet connection is poor, and you have to run it from the cell one again and again. Okay? Third thing is important about the Colab is it gives you the CPU access of the Google system. So if I show you, sorry. No. If I show you that this section should be connected. Okay, this is not sign in right here, but it should be says that you are connected. Okay, and it gives you the connection on the Google. So it will save your file up to 8 hours only. So if you are keep running and open your laptop up to 8 hours, you can run it in a single time. But if you close your laptop and run it on the next day, you have to run it from the cell one. Okay, understand? When it gives you the model, you can download it. It has an option. It is already downloaded. The downloaded models have multiple things, relaxed version, unrelaxed version, or the more iteration limits. Some of the students do not have the relaxed versions of the PDBs. They can use the iteration limits. Okay, it says 0, 1 PDB, 0, 2 PDB, and 0, 3 PDB like this. So you can use the model 1 on that. After model 1, you can evaluate that model on different web servers. If you write on the Google evaluation of 3D structure model, you have number of multiple servers, but the most reported ones are the PROSA and the SAVES. So we use PROSA here, and you can utilize number of tools in that one server, like Ramachandran plot, like red plot, and local energy minimum. Okay, so this is the server where you can use multiple tools in the one server. Okay, fine. So we end the workshop here. I hope you all noted email address and contact numbers in the sheet. Yeah. Maria. And okay. Thanks, Ms. Atya and Ms. Maria for facilitating this workshop. Professor Dr. Zaveer is not here to conclude this workshop. So on behalf of him, thank you so much for your time. I hope you gained very much from this workshop. You can email me. I'm Dr. Ruz. I have an email address written on your manuals. And you can contact Dr. Zaveer directly as well. Thank you.
एक्सप्लेन किया आप लोगों को पूरा कोशिश मैंने की कि मैं करूँ और ज्यादा होगा हाँ पता नहीं सबके रेफरेंस से ज्यादा पार्टिसिपेंट थोड़ा कम और यहाँ पे